Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled, Establishing an Expert Network for Your Radiopharmaceutical Drug Development Program. My name is Sonia Hunt and it's my pleasure to be your X-Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. Now, it is my pleasure to introduce you to one of your speakers for today's event, and he will also be your moderator. And he is Dr. Cyril Berthet. He is the head of Pharmaco-imaging and the theranostic platform at Oncodesign. He holds a PhD in molecular and cellular biology from the University of Lyon. In 2002, he joined the mouse cancer genetics program at the National Cancer Institute in Frederic, USA, as a research associate. In 2007, he joined Oncodesign as a project leader and managed strategic partnerships in biomarker and drug discovery. Now it's my pleasure to pass over the mic to Cyril to introduce you to our speakers for today's event. So Cyril, when you're ready, you may begin.
Thank you, Sonia, and it's my pleasure to be the moderator of this session and this webinar. We will try to be as interactive as possible and not be too long and keep times for the Q&A, so feel free to ask all the questions you would like about this expertise in the nuclear medicine fields. We will start with different speakers, and I will introduce first Dr. Meddy Alaoui. He's the CSO of Covalab. He did his PhD in biochemistry at the University of Lyon. He worked as a postdoc at the Cancer Center in Lyon and the University of Cambridge on the development and validation of cross-linking technology. He has so good expertise in the bioconjugation field. In 2018, he joined Covalab to lead and develop the COVID platform applied to antibody fragments. Next, we will have the pleasure to have Frederic Boschetti. He's the CEO of CheMatech, one of the leading companies in chelating agents. He obtained his PhD in chemistry from the University of Burgundy in Dijon in 2003. And in 2005, he founded CheMatech, a company, as I say, specialized in chelating agents. So he's involved in many research programs and has a strong expertise in the field of radiochemistry. And finally, we will have the pleasure to have Andreas Kluge, the general manager of ABX-CRO. He has over more than 20 years of clinical research and development experience. He also was a founder of CEO of ABX GmbH. He is the author of numerous patents and publications in the field of nuclear medicine, neurology, and infection immunology. He has a very strong expertise in clinical development of radiopharmaceuticals. Will be our different speakers, and I will start to introduce our DRIVE-MRT, which is the global offer for as an expert network for your radiopharmaceutical development, drug development. So we will start with the first polls just to have an idea of who is attending and what is the level of experience of our audience. Please, Sonia.
Thank you very much, Cyril. I will launch our first poll questions, and there it is, audience members. Please go ahead and select the below, whichever one applies to you. You have about 45 seconds to complete this simple poll question, and then I will close the polls and share the results with everyone, and then we'll get right back to Cyril. Okay, that looks like it's a majority of our members in the audience have cast their votes. Just a few more people there are just, looks like you're about to cast your vote. If you could just go ahead and complete it now, and then I will close the polls. Oh, perfect. Thank you so much, everyone. That's a great majority of our audience participating. Thank you. I'm going to close the polls now and share the results. Okay, so the results are here. We have 41% of our audience, I am a newcomer in the field. Then we have 44% that are saying, I know my way around. And then we have 15% that are saying, I am an expert. So thank you, everyone, for participating in that first poll question. Now back to you, Sarah.
Thank you, Sonia. So for all the newcomers and the ones that are not fully familiar with the radiopharmaceutical fields, really feel free to ask the questions, and we will try to answer as much as we can to all these questions. So the DRIVE stands for Drug Interactive Drug Discovery Molecular Radiotherapy. We really would like to have a global offer starting from chemistry to clinical development as a premium nuclear medicine solution in oncology. We have different companies here that manage to work together as an alliance of specialists in the radiopharmaceutical fields. And this specific development require dedicated expertise in nuclear medicine. So Covalab and Meddy Alaoui will present some specific development on antibodies, antibody fragment, and bioconjugation. Then we will have CheMatech that will introduce the lead optimization, the custom design and synthesis of chelator. I will take then the lead from the preclinical proof of concept, or the studies that you have to do in the preclinical development. And finally, ABX-CRO will introduce what is needed and required for the clinical development. So this is global offer, start from target in validation up to clinical development. And this is the short introduction. I won't go into further, and I will pass on to Covalab and Meddy Alaoui to introduce his company and expertise.
Great, thank you for the kind introduction. So Covalab was founded in 1995. We are experts in the field of antibody engineering. Our headquarters are based in Bron, close to Lyon in France. We've got also our own animal house in Dijon, and we have more than 2 decades in immunotechnology expertise. And most of our parts, we have a fully independent antibodies engineering platform in order to develop various type of antibodies. So we are at the beginning of the part of DRIVE-MRT in order to develop new druggable compound. And for that, I will present you my next slide, our antibody development process fully integrated in Covalab. So first, we can start with the immunization parts that could be conventional or based on DNA that I will present in the next slides. And after, we can develop those antibody by using polyclonal or monoclonal antibodies. And for the next step, we can engineering these antibodies such as full length, but also with fragments such as fed fragments, LCAV nanobodies, also known as VHH. Just going back, please. So we can work, just as I said before, for chimerization and humanized antibody. And last but not least, we also can perform bioconjugation process through our COV-ISOLink technology. So now I will present you why and using DNA immunization and how we can help in your process. So DNA immunization, to be also said DNA vaccination, is an in vivo transfection. To make a long story short, it's the sequence of the protein of interest inserted into a plasmid and simply injects into mice, rabbits, or other animals without any specific equipment such as electroporator or gene gun. It's a very simple injection that allows the protein expression at the cell surface, which will be displayed for the immune system. And one of the main advantages of this method that the antigen will be displayed as a conformational parts, not as a linear one, but as conformational epitopes that could be required from specific application and to develop very good antibodies. This could be very interesting too for 3D complex targets, such as transmembrane protein for cancer receptors, for example, or for some post-translational modification. That leads to get very good antibody with very high specificity and high affinity that could be required for MRT development antibodies. So in my next slide, I will present you how we can make a very small antibody, because depending of your final application, full-length antibodies such as conventional IgGs maybe not the best tools. For some of them, VHH fragments, also known as nanobodies, have all a size of only 15 kDa. It's 10 times smaller than conventional AGG and can bind hydrogen and with better permeability. But for DRIVE-MRT application, maybe one of the most advantages is fast clearance, because due to its very small size, 15 kDa, it could be removed from the renal clearance out of the body very fast, and by using radio-labeled compound, that could help to get better results. So we have in my next step, so global projects at home that could be from naive library from Chemelids and Llama. We are able to perform the whole project process to the high-scale production of the VHH. For the next slide, I will present you one of the most important parts also for the antibody development, which is the bioconjugation part through our co-isoling technology. Even if you develop maybe the best antibody that can spend many years to do it, if you make random conjugation from at the end, for example, with a chemical reaction, you can lose the antibody affinity and specificity. To avoid that process, we developed a new enzymatic conjugation based on a Q-tag that you can see in purple on the antibody. This Q-tag is containing a glutamine substrate from the enzyme. And by using the enzyme, which is called transglutaminase, the enzyme will cross-link to this Q-tag any specific compound from radiolabeled element, chelators, or different drugs, for example. And that is very important on this part. It is control and site-specific cross-linking. And this is suitable for therapeutics such as MRTs, ADCs, but also for diagnostics, radiolabeling, and also for R&D with dyes. One of the most important things with this co-isoling technology, it's available for every MABS format, such as PEB, single-chain SCAV antibodies, and even VHH. Because of this very small size, VHH are very hard to cross-link without any loss of immunoreactivity. Based on our technology and with the Q-tags, you can easily cross-link any chelators for MRT application with VHH without losing any specificity. That could be very convenient for the DRIVE-MRT process. So to summarize our part in DRIVE-MRT in my last slides, so we can start from the target identification and give a hand for based on innovative immunization process such as DNA immunization I presented before. We can also work on those antibodies with antibody fragment, for example, and to the end, developed the bioconjugation by our co-isoling technology for specific cross-linking even with antibodies fragment. And now I will leave the floor to Kematec to present their own technology. And of course, I'm here for any question at the end. Thank you.
So thank you, Meddy. So good morning and good afternoon, or good afternoon, everybody. So I'm Frederic Boschetti, CEO of CheMatech. So I founded the company in 2005 as a spin-off of the University of Burgundy in France. So our company is specialized in the design and production of chelating agents for nuclear medicine and molecular imaging. So currently we have a portfolio of more than 1,000 clients all over the world. So we generally sell our products directly to the customers, but we also have a network of distributors to assist us in different countries such as Japan, India, China, Russia, or South Korea. So we are in this market since more than 15 years now, and we are one of the major actors in the field. So the chelate make the difference. Actually, this is my favorite expression. It reflects the importance of the chelator in radio tracers development. It was actually the title of a publication of Professor Mackey in 2011. So when designing a radio tracer, you need a biovector, such as an antibody, a peptide, or a small molecule, a linker to attach the chelator, and a radiometal. So depending on the radiometal, you can use the radio tracer for diagnostic, for instance, with gallium-68 or copper-64, or therapy with lutetium-177 or actinium-225. So as you can imagine, the main role of the chelator is to trap the radiometal. So you have to adapt the chelator toward the radio element in terms of coordination sites and side, and in order to get the most stable complex and to avoid the in vivo release of the metal. but you also have to play with the chelator to get optimized radiolabeling and reach high specific activity. So in addition, you have to find a chelator allowing radiometallation conditions compatible with the stability of the biovector. For instance, for a full antibody, you have to perform the radiolabeling in mild conditions in terms of temperature, so you cannot heat above 40 degrees, and in the appropriate pH conditions. So the ideal case will be having a chelator allowing radiometallation reaction at room temperature under mild pH condition and with high specific activity. And this is a real challenge. But beyond radiolabeling reaction, the choice of the chelator can have an impact on the properties of the radiotracers in terms of pharmacokinetic and biodistribution. So many parameters can affect the in vivo properties of the tracer, such as the global charge, the geometry, and the lipophilicity of the chelator. The radiometal itself will have an impact on the biodistribution. So it is well admitted now by the scientific community that the choice of the chelator is of high importance in the design of a radiotracer, and that the chelator don't act as an isolated species and is not only here to trap the radiometal. So last point, which has to be considered, is the choice of the grafting function. Indeed, you have to add to the chelator a reactive function allowing the bioconjugation reaction. So we'll not use the same reactive group if you want to attach the chelator onto a cysteine, lysine, or onto a bioorthogonal function. Moreover, can you go back, Cyril, please? Moreover, the choice of the linker will also have an impact in vivo property of the radiotracer in the same manner than for the chelator. So what I wanted to highlight here is that you cannot treat and optimize the different components of a radiotracer independently from each other. You have to find the ideal combination between the chelator, the radiometal, and the biovector in order to get optimized properties. And of course, in CheMatech, we have the expertise to assist our clients in the choice of the most adapted chelators. Next slide, please. So here in CheMatech, we are able to play with all the factors and can have an impact on a radiotracer properties. So for instance, we can play with the size of the chelator by using a DOTA derivative or a smaller one like NODAGA. We can also modulate the number of coordination functions and the charge of the complex by using a DOTA-GA instead of the DOTA. We can also play with the type of reactive function for the bioconjugation. Two of the most used are the NHS and NTS groups, which target lysine or more generally primary amine. But if you want to attach the chelator onto a cysteine, you will have to use a Meddy group. You can also use a click chemistry function like this Bayesian DOTA, which is often used for the site-specific bioconjugation of antibodies, like in the Covalab technology, for instance. A spacer can also be added, such as a PEG4, this PEG4 linker. Addition of such moiety usually improves the in vivo retention time of the tracer. So beyond this DOTA and NOTA derivative, we also produce other chelators such as DFO for zirconium-89, RESCA for aluminium F18, or THP for gallium-68. We also produce other small molecules such as PSMA targeting agents precursors. And as you can see, some of these chelators are available in GMP quality. Next slide, please. So at CheMatech, we offer a full service for the production of chelator for nuclear medicine. So we offer custom design and synthesis of chelator from the conception to the preparation in bulk. We are able to produce from the gram scale to the kilogram scale of a compound. We have a GMP unit dedicated to the production of GMP starting material and intermediates. This unit is equipped by automotive glass reactor from 500 milliliters to 30 liters, industrial water vapor and flash chromatography. As I told you, we already have some GMP chelators available, but we also do custom GMP synthesis. We can produce from several grams to 1 or 2 kg of compound. So for this activity, we have the authorization from the French FDA. So we are currently expanding our GMP facilities, and the new building will be ready by the end of the year. So as a chemical supplier, we have a catalog of products with around 100 chelators. Here you can find most of the chelators involved in the preparation of radiotracers. We also participate in joint research program in order to keep a research activity and develop new solutions for our client. So in summarize, we can develop and produce both R&D and GMP chelators from grams to several kilos. So now I will let Cyril from Oncodesign talking about preclinical evaluation of a radio tracer.
Well, thank you, Frederic. It looks like there's a poll question, so I'm going to launch that full poll question before we hand it over. So let's launch poll question number 2. Okay, there it is, everyone. Just like before, it's done in real time, and your participation is strongly encouraged and very much appreciated. And the question we have for you for poll number 2 is, do you have a project in the nuclear medicine field? So please pick one of the below: ongoing imaging diagnostic project, ongoing therapeutic theranostic projects, or future plan to develop such projects. Okay, that looks like it's half of our audience. Okay, come on, the rest of you, please go ahead and pick one of the below, and then I will close the polls and share the results with you. Oh, fantastic. That looks like it's a majority of our audience. Leaving a few more seconds for those of you who have not chosen as yet. Okay, that looks like it's everyone. So thank you, everyone, for participating. I'm now going to close the polls and share the results. Okay, so here's the results. We have 16% are saying ongoing imaging diagnostic projects. Then we have 48% that are saying ongoing therapeutic theranostic projects. And then we have 36% saying future plans to develop such projects. So thank you very much, everyone, for participating in poll number 2. Now back to you.
Thank you, Sonia. So now I will talk more about the preclinical developments of the nuclear imaging on molecular radiotherapy. At Oncodesign, we can start from the bioconjugation and taking the compounds, the chelating agent from Kematec and biovectors from Covalab or any vectors that you may have, and perform the bioconjugation and radiolabeling. Then we will have a steps of binding assessment on biological samples. It's very important to evaluate the true binding of your compound. It can be on the cell models or it can be on tumor biopsy or whatever. And the key steps is the biodistribution. How much of your compound is going to the tumor? This is really a crucial steps. And when you have this data, then you can move to evaluate the efficacy if you have a therapeutic compounds and start to think about your first-in-man administration. We have different agents, as it has been said before. You can work on therapy with lutetium, indium-131, actinium-225, or you can work on diagnostics part with different isotopes, such as zirconium-89 for antibody, gallium-68 for short PK compounds like peptide and so on. And I will go to different steps. For the optimization of bioconjugated compounds, Frederic has already mentioned a couple of important parameters, and you have to define if you want to do a random versus a site-specific technology, which isotope you want to use, the condition of the bioconjugation. It also depending on your vectors. If you're working on small molecules or often antibodies, you may not be able to use the same condition of bioconjugation. The impact on the radiolabeling. What specific activity you may reach, which radiochemical purity, radiolabeling yield. You have a lot of parameters that you may need to optimize in order to have a good compound at the end. And impact on stability, radiolysis, affinity. If your bioconjugation modify the affinity of your compounds, notably if you have a small molecules or nanobodies, it might not be always easy to not impair the affinity, and it has to be checked to make sure that your compound has still a good selectivity and good affinity. And at the end, the impact on biodistribution. Frederic showed you some published data that the chelate may change the output of your compounds. So what we will propose is kind of a toolbox to really optimize your lead compounds. And once we have the different chelating, the different linkers options, we will evaluate the radiolabeling and the in vitro binding selectivity. And the key steps, as I say, is the in vitro biodistribution. I like to say that our goal is to reach at least 5% of the injecting dose in the tumor, and this is really a good starting point. If you are below that, it can be more challenging for an efficacy treatment. For diagnostic, this is manageable. So when we are talking about binding affinity determination, you have a first step is to do the binding on your selected model. It could be an internalization assay, could be done on lysate, could be done on section of your preclinical models. There are different ways to do it, but the goal is to evaluate the specific binding versus the non-specific binding. And you can even go to define the KD of your compound and the Bmax, so how much of your compound is going to be really bound to your target. But once this is done, and you have done all your preclinical developments, one important issue that you may have is to enroll your patients for your phase 1. And how many of these patients in the indication of your of interest will truly bind, truly have an uptake of your vectors. And here we can propose a binding, an ex vivo binding assay on tumor biopsies from different patients. So here, for example, you have some interest in pancreatic cancer, and we can do an assay on 10 different biopsies. And if you have a good binding on these biopsies, on most of them, that you know that you can select all the patients to be enrolled in your clinical trial. If only 10%, or here on the picture, only 50% of the biopsy you have a binding, you may need to refine and to identify a new biomarker to subdivide your population and really enroll the patient that has a good chance to have an uptake in vivo. So this assay is really powerful to avoid mis-enrollment of patients in your clinical trial. Once you do the bioconjugation and you have your compound that is validated, then you need to do the radio labeling, and it's kind of go-no-go in your steps. And when everything is done, then you can really do a true biodistribution study. And the biodistribution study is basically to select your models and to really define how much of your compound is going to the tumor or to your organ of interest, depending on your target expression. We are mainly focusing on oncology, but of course for diagnostic, it can be any other targets. It could be in the brain, it could be in the heart, or whatever organ your target is expressed. And this biodistribution requires expertise from chemistry to data analysis. And if you are interested in the therapeutic aspect, then you need to evaluate the efficacy of your compound. So here we just show two examples of one compound that is radiolabeled with lutetium-177, and you have a complete response of your tumor. If you play with the dose and the schedule, here the same compound that half of the dose has been injected but twice with 7 days apart, then we observe still a complete response, but a delay in the relapse. And this is really something that we can play with. There are different parameters that are crucial in this kind of assay, is of course the dose, the schedule, and the specific activity. As you need to understand that not 100% of the molecule are active due to the decay, due to the radiolabeling with the isotope. A lot of compounds are actually cold and are not active, and even can saturate your target and limit the efficacy. So we have to play with this ratio of cold compound versus radioactive compounds to define the best condition of the efficacy related to the specific activity, different schedule, and the correlation with the tumor uptake. And once we have define the best parameter, we can play on the biodistribution. We can do the biodistribution, the full dosimetry, so how much of radioactivity is dosed in each of the organ, the expression of the target, and different biomarker of mechanism of action. And to go beyond and move to your clinical indication of interest, we can perform efficacy in PDX model, patient-derived xenograft models, so that are more relevant to the patient than the standard syngenic or xenogenic xenograft, combination with standard of care. And as we have more and more experience in clinic, people are asking for combination in preclinical settings with standard of care or with immunotherapy. We know that there is a strong synergy of radiotherapy and immunotherapy. And we can also evaluate different isotopes. We know that alpha or beta isotope may have different therapeutic impact, and this is something that we can play with. And it has been defined from the start in your program to define the best skeleting agent you want to use. And before passing on Andreas Kluge, we will have a third poll to finalize that. Sonia?
Okay. All right, thank you very much, Cyril. Yes, it is. It's our third poll question and final poll question for this webinar, and there is a question in front of you. Okay, so please go ahead and choose from the below. Which radioisotope would you favor for your MRT project? I'll give you 35 seconds to complete this simple poll question. We have Actinium-225, Lutetium-177, both, or would like to test other isotopes are your options. Okay, that looks like it's the majority of our audience members participating in that final poll question. I'm now going to close the polls and share the results with everyone. Okay, let's take a look at the results. Okay, so we have here for actinium, we have about 17% of our audience members that are saying yes to that. Then we have 13% are saying lutetium, 177. And then we have 43% of our audience members that are saying both. And then we have 26% that are saying would like to test other isotopes. So thank you everyone for participating in our final poll question. Now back to you, Cyril.
Thank you, Sonia. And so I will now pass on Andreas Kluge. Andreas, have you joined us?
Yep, I do.
Thank you.
Hello everybody. Thank you for the presentations and for the introduction, Cyril. So ABX-CRO, some of you may have heard the name. ABX-CRO is an independent international full-service CRO, privately owned, founded in 2002, specializing in nuclear medicine, and nuclear medicine, which relates mainly to oncology, more infrequently to neuroscience, and of course always to imaging. We are headquartered in Dresden, Germany, and have local offices in Australia, Spain, Switzerland, South Africa, and a small one in the US, and prepare offices in other places of interest for clinical research like India and South America. We are at the moment a team of 100 people. And our clients, we boast big pharma, small companies, other CROs frequently. We have a rigorous quality system. We're ISO 9001 certified, and for our software developments, we are ISO 13485. The company has been in long-standing research collaborations with the EINM. We have been part of implementing multi-center clinical research, and as such we have run global clinical trials with multi-center PET imaging. So in fact we have developed the methodology together with the opinion leaders in the field. The US clinical trial network of the SNM was inspired by the respective initiatives we launched in Europe. Yeah, so next slide please. Cyril. Okay, yeah. So our services, it's more a pilot's view here. We especially are interested in scientific consultancy, study design. The colleagues have covered the early stages of development. We particularly specialize in designing your studies, considering the imaging requirements and the information you want to get out of the imaging as part of the clinical study. So we're a CRO which really integrates the imaging part of the CRO work with the clinical operations, because clearly in order to get the most out of your clinical images, you better closely ally the clinical aspects with the imaging requirements. So to this end, we procure you with feasibility, site selection, setup, having in mind, of course, the patient, the patient source institutions, but also, of course, to select suitable imaging institutions which have the requisite camera setup, because many institutions which may be patient-wise interesting have no adequate state-of-the-art imaging equipment. So we have to build composite sites, which is part of our special know-how. So the technical feasibility for camera equipment, we bring up to speed cameras by interacting with vendors and manufacturers in order to really bring the camera performance and normalization to a point which allows really quantitative comparability in a multi-centric setting, which is in radiopharmaceutical development not only necessary in late stage but also in phase 1, which is typically phase 1 multi-center setting in order to get acceptable recruitment recruitment durations, which still is not always easy in nuclear medicine studies, because you have the classical situation that the oncologist and the nuclear medicine physician, two disciplines, really have to pull on one string in order to make the trial happen. And that's often a lot is promised, and if you come uninformed to a site, and then the performance is disappointing, which of course is our main sorrow as a CRO, to make you happy with recruitment rates. So regulatory setup involves in radiopharmaceutical studies the usual clinical trial authorization. You know that in many countries, in addition, we have the radiation safety track. So there are separate institutions in France, in Germany, and in many other places, which deal just with the radiation safety permission for a clinical trial, which is completely distinct for the pharmaceutical clinical trial authorization. So we provide you with a complete package. Our clinical operations cover phase 1 to 1, including phase 4, although phase 4 is very rare in radiopharmaceutical development. As key part of what we do, our dedicated image core lab performs really the camera qualification, and of course the biodistribution study I'm going to elaborate further, and dosimetry, which is part of any development of new radiopharmaceuticals, data management, biostatistics, and the usual stuff, medical monitoring, pharmacovigilance we also offer. Next one, please, Cyril. So I think at the core of any radiopharmaceutical development is the investigation of biodistribution, and to analyze biodistribution for dosimetry from a safety perspective. In terms of this is mainly required for any radiopharmaceutical diagnostic or therapeutic, and if it comes to therapeutic radiopharmaceuticals, you also would like to know how much uptake and finally absorbed dose ends up from an injected, let's say, 5 gigabecquerel lutetium dose in your tumor. And we have discussed previously that the selection of the right chelator, the right linker, the right nucleate really can direct a fraction of your injected dose to the tumor. So Cyril has mentioned the specific activity, in other words, the substance dose which you give for the predefined activity dose, let's say 5 gigabecquerel. You can have anything from 1 to 5% ending up in your tumor as a function of the mass dose, considering off-target binding and the different equilibria for radiolabeled and non-radiolabeled compound, especially for antibodies, but this is the mass dose is important, but also for peptides and also for small molecules, as becomes increasingly clear. So any refined, rationally designed development program for therapeutic radiopharmaceuticals should address the question of what is the suitable specific activity, which is not obvious and which cannot be extrapolated from preclinical data. That there is an effect of a mass dose effect on tumor uptake percentage can, however, be addressed in a preclinical study, which gives you an indication whether this problem has to be tackled. So this is how the dosimetry workflow looks like in the QDOS software, which is a clinical grade software, which is also marketed and developed and produced by ABX-CRO. You load your data in, you segment your organs. Typically, you have planar images in which you identify your organs at the different time points, and you then plot the time activity curves, and you integrate the totality of these integrations in that organ, considering further standard neighborhood relationships from so-called anthropomorphic phantoms, which assume a standard anatomy and then you get this plot, which is very slow, right, left, bottom, which gives you absorbed dose estimates for a standard anatomy, person, male and female, for a total list of 26 organs, and a total absorbed dose normalized for normalized for 1 megabecquerel typically, or for the dose to be given. So next one. So our clinical trial experience, you always know everything is secret in pharma, so we did more than 40 trials in the last 15 years, covering biologicals stemming from even radioactively labeled cells, including monoclonal antibodies, peptides and small molecules, radionuclides, therapeutic, Yttrium-90, iodine-131, lutetium, different radiums, to name a few. We also do diagnostic development these days, mostly gallium-68, F18, and but also iodine-124, which is a suboptimal PET tracer, more recently zirconium, which might be a topic for you. And we also do so-called pharmacological studies with radiopharmaceuticals, which is typically used in CNS tracer development to assess receptor occupancy, or the relationship between plasma level and this receptor occupancy in order to achieve a rational dose finding in the target species, which is man, where you have the blood-brain barrier. So also C14 is a tool for so-called ADME studies, which gives you the fate of your compound. That's also something, but that's beyond the proper radiopharmaceutical development. It's here just a research tool for development of of drugs which are not intended to become radioactive. So we have, as I said, global clinical operations, including US, including the EMA area in South Africa and Australia and New Zealand. We provide global regulatory support for pharmaceutical and for the radiation safety aspects, and we have several products we have developed are meanwhile authorized. Next one. So I think this is the moment to hand over to Cyril again. Maybe Cyril, you jump on the drive. So the DRIVE-MRT really is here presented as a total solution which helps you to bring really whatever your stage of entry is, whether you're in the discovery stage, whether you have a new target, where we can help you in the DRIVE-MRT consortium to identify biological ligands and pharmacophores. We can radio label them with the colleagues of Kematec. We can do the preclinical evaluation, and we can take it further in clinical development, taking you by the hand to talk to regulators at the global level and run your trial, analyze the data, and we have the ambition to be your partner, as you feel as if we're part of your organization, because it's clear that the amount of specialist expertise is quite rare on the market, and we hope this is an interesting offering for anyone developing radiopharmaceuticals.
Thank you, Andreas. And clearly, this is the spirit of this alliance, is really to have a strong collaboration for your project, and really help you to move fast in the different fields from chemistry to clinic. And here is just a summary of the package from radiochemistry, bioconjugation, and even antibody engineering up to the clinical trials. We feel we have a strong synergy between the partners, and that can really help to push forward your project. And now we'll be ready for the Q&A section, and feel free to have different questions, and we'll try to be as interactive as possible.
Okay, well, first of all, thank you very much, Cyril, Meddy, Frederic, and Andreas for that very insightful presentation. I hope everyone enjoyed it. And now, just like Cyril said, we are now going to begin the Q&A portion of the webinar. And for that, hi everyone. And for that, I would like our speakers to put on their webcams. And I'm going to just remind the audience that you can send in your questions by using that questions window that's located on the right-hand side of your screen. And we'll try to address your questions during this time that we have together. Now, I'm going to pass it over to Cyril to lead the Q&A. So Cyril, just invite me back when you guys are done. Thank you.
Thank you. Yeah, we have a first question for Frederic, actually, for Kematec. So for your presentation, may I ask you if it is possible to perform external audits on your facility in case we would like to express an interest to procure chelators?
Yeah, of course we are open to audits. So we have very frequent audit from our clients. So we will be more than happy to welcome you in Kematec and to show you our facilities and our capacities and what we can do about GMP synthesis of chelators. So no problem. We are open for audits.
And then I have a question for myself. When someone is asking if Oncodesign is open to accommodate a developed radiopharmaceutical outside of Oncodesign to be tested in our facility. Yes, of course, we can receive compounds developed from outside. It's manageable usually with lutetium or actinium. Of course, if you want to do gallium, will be more challenging. But we can either transfer your methods of radiolabeling and do the study, or if the half-life of your compound is long enough, we can receive it. Is it possible to perform the experiment yourself? No, in that case, it will not be possible. You're welcome to come and assist, but that will be our technician team that will run the studies. And do we have other questions here? I can see. For the last one, another question for novel radio diagnostic and for platform-based diagnostic, what preclinical safety data are needed before humans can be dosed? So this is a question for Andreas. So what preclinical safety data are required before human?
Yeah, well, thanks for the question. I think it really depends on what type of agent we are looking at. You may be aware of the so-called microdosing concept and the microdosing guidelines. So this specifies essentially that 100 microgram mass dose, or alternatively a factor of 100 from the no observed adverse effect level, the null level, can be considered a so-called microdose, whatever is lower. And for this, you're required to provide just a so-called extended single dose toxicity study in a single species, which is an investment of between 50 and 100,000 euros, and which is doable with a overseeable amount of compound and contrast to full-scale tox studies at this stage, which for a conventional tox study with the multiple dosing in two species, you have to have at least a 5 to 10 gram compound at hand, which typically is in early stage of development a problem. So for a microdosing study, you need this one extended single dose toxicity study, which allows also in the US to give you up to 5 of such doses repetitive if you want to do some intervention in the meantime, provided that the half-life of the compound can be assumed to really have no remaining compound in the system. So if the intervals between these dosings, you can go up to 5 repeat doses as a microdose with a single dose toxicity study. Is that answering the question?
Yeah, thank you. And we have a new question for Meddy. Is DNA immunization working for cytoplasmic protein?
That's an interesting question. So that's not that obvious, but yes, we developed a new kind of plasmid that allows the expression of intracellular protein at the cell surface in order to develop DNA immunization and conformational epitopes by using DNA immunization, cytoplasmic and even nuclear protein.
Okay, thank you. And oh, again, a question for Andreas. Can the QDO software is suitable for alpha emitters?
Yeah, very much so. Thanks for the question. I mean, the QDO software is, the other software out there is Olinda. There's all current nuclides implemented, whether it's lead-212, actinium, and all these alpha compounds. There's one remark I would like to make. We have the tendency, and this is especially true for actinium, and I appreciate that the audience is heavily active in actinium. Everybody probably thinks of the supply chain issues, or not yet, but this is to be a big issue. I would like to make the following comment, and we have the chelating agent expert chemist here. So the general expectation is that, especially in multi-alpha decay nuclides such as actinium-225, the binding energy of the actinium in a DOTA or whatever you choose is probably less than the first alpha decay. So the point is that in multiple alpha decays, likely your daughter alpha decays will not take place inside the chelator. So these nuclides will, depending on where the primary place of decay is, will likely have their own fate inside the body. This is different, for instance, for a bone-seeking alpha like radium-223, which is licensed for therapy of, as you know, this Xofigo from Bayer. Here, likely the uptake in the bone matrix will lead to a more localized persistence of daughter nuclides. But if we're starting to treat soft tissue metastasis, which is, for instance, in many fields of interest, then we have to assume that the daughter nuclides will not stay in the vicinity of the primary decay. In order to tackle this problem, or at least to conceptually understand it, ABX-CRO, in addition to QDOS, so QDOS does that, using conventional dosimetry models, but in order to assess really the impact and the particular dose exposure of organs, assuming that the daughter nuclides are not staying where the first decay took place, we have a research-grade dosimetry software called IDAC Alpha, which models the fate of the daughter nuclides, and we can. There's a paper out which I am happy to send anyone interested in the field. So please feel free to reach out to us. IDAC Alpha estimates, for instance, for radium-223, which is a bone seeker, that the absorbed dose to liver and kidney are calculated under consideration of the daughter nuclides. Even in a bone seeker is about 20 to 30% higher than with the conventional dose algorithm, which is implemented in QDOS and also Olinda. And this proportion is likely to be higher in soft tissue seeking constructs with, for instance, with actinium. Conversely, however, in the bone seeking situation, radium exposure is 20% less than conventionally estimated. So that is why you typically do not see relevant bone marrow suppression with radium-223.
Yeah, thank you, Andreas. We clearly understand that determining the dosimetry of alpha emitters can be quite complex, and you really rely on strong expertise, as you may have. We have a good comment. So impressive combination of expertise. Thank you. With a question, are we supporting studies in United States as well, both on CMC and clinical regulatory? So I think for the chemical and preclinical work, it can be done anywhere. And of course, we have many clients in US. And for the clinical and CMC, Abxero has all capacities to run studies there. And I will let you comment, Andreas, on that specific point, to manage clinical studies in US.
Thanks for the question, and the answer is of course yes. We cannot do everything ourselves. We're a team of between 90 and 100 people, so we rely on preferred partner with whom we have established long-standing collaborations, and which we can put together in a one-stop package as inside DRIVE-MRT. So essentially can assume that all capable radio expert people in the world know each other and are happy to work with each other. And of course, we are really inspired, and I think I can say that for me, but also for the colleagues. We are really feeling that here a new category of medicine is about to take over a relevant part of oncological care, and I think this has been perceived by a lot of physicians already, certainly by the investment community, and I think anyone working in the field, I hope that's true for our audience, is also a believer. So we can put together teams which work. And in the US, absolutely, from CMC to regulatory, to run the trial, what you need.
Yeah, we all believe that radiopharmaceutical have a strong perspective in the future, and this is also why we are all here to really help everybody to push the program faster. We have a question for Frederic. I think you have answered this question in your talk, but might be good to refresh everyone. What is the typical scale up for your GMP production, and usually what is the amounts that people are requesting for I would say for phase 1?
Yeah, good question. It depends actually. It depends on if the chelator is for diagnostic therapy but for the phase 1 you need I would say few hundred grams typically. So below the kilogram of product. So usually we make batches of around several hundred grams of product, and usually it's enough to perform clinical trials. So this is small quantity.
Yeah, even for the bioconjugated antibodies or vector, we try to have dedicated partners, and we try to work on that to also offer a specific, the CMC that is dedicated to radiopharmaceuticals, because you don't need as much as for a standard therapy, and it's important to produce what you need and to have a CMC that is adapted to radiopharmaceuticals and that is clearly different in terms of total amount that you need from standard therapy. And I think we have a last question, unless there is other question that's popped up. One is related to combination efficacy studies. Can you compare an immunotherapy with MRT versus immunotherapy with standard external radiotherapy? This is done in question for Preclinical. So yes, we have access to an external radiotherapy system as well, a small animal irradiator, so we can do such comparison. And the effect can be different when you combine with immunotherapy. This is a very interesting field of research, and we can see there is more and more data that are published by different teams with combination of immunotherapy and MRT or radiotherapy. The goal of the radiotherapy, whatever it is external or internal, is really to transform and to modify the tumor environments to have more inflammation and more immune cells within the tumor, and that you can then target with immunotherapy. And there are very strong synergy between these two components. So this kind of research and settings are very interesting to be developed, and we have experience in the field, of course. I think we are at the end of the session, and thank you all for attending. It has been a pleasure, and we hope that we'll be in touch soon with everyone.
Okay, well, first of all, thank you very much, audience members, for those questions again, and thank you very much to our panelists. We have reached the end of the Q&A portion of the webinar, and if we could not attend to your questions, the team at Oncodesign will follow up with you after this presentation. Now, if you have any further questions, there's a bunch of email addresses and phone numbers here that you can connect with our speakers, so please go ahead and utilize them. They'll be very happy to assist you. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will also be popping up on your screen. Your participation is strongly encouraged as it will help us to improve on our further webinars. Now in a few seconds, I'm going to send you a link in your chat box. You'll be able to view the recording of this event with that link and also share this link with your colleagues once they register for the recording as well. So I encourage you to do that and get even more out of today's presentation by downloading the supporting material that is available under the Handouts tab, and we have a PDF there for you, the DRIVE-MRT Expert Network for your radiopharmaceutical drug development program. So please go ahead and check that out. And once again, let's please say thank you to our speakers, Meddy, Frederic, Andreas, and Cyril, for that very insightful presentation and for answering all your questions. So thank you very much, gentlemen.
Thank you. Welcome.
Thank you.
Thank you. Thank you. Perfect. We hope you found this webinar informative. It has been our pleasure to be actually, it is my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Thank you, everyone. Bye, everyone. Thank you, Frederic. Bye, Meddy, Cyril, Andreas. Thank you.
Bye.